Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International IGCSE Biology Paper 2BR for January 2023. Let us begin with the first question. So here they say, read the passage below, use the information in the passage and your own knowledge to answer the questions that follow. This passage is about antitransparents. They say antitransparents are substances applied to plants to reduce water loss. There are three types of antitransparents: film-forming compounds, stomatoregulators, and reflective compounds. The first type of antitransparent contains film-forming compounds. These compounds are oils or waxes that are mixed with water to form an emulsion. This emulsion is then sprayed onto the leaves of plants. The emulsion forms a physical barrier that covers the stomata. The film is designed to prevent water loss but allow gas exchange. Studies have found that the film-forming antitransparent is effective in increasing yield and increasing photosynthesis in drought conditions. The increased leaf turgor in plants sprayed with film compared to control plants shows that the film decreases water loss and maintains turgor in drought conditions. Film-forming antitransparents are used in horticulture to reduce water stress. This can occur during storage, transport, and planting of container-grown plants. Application of the antitransparent before transplanting helps the plant survive until the root system is established. The film-forming compounds are used to reduce drying out in rolled turf and in transporting flowering plants and trees with root balls. The second type of antitransparents are stomatoregulators that reduce the size of the stomatopore. Stomatopore affect diffusion as well as transpiration. Therefore, photosynthesis and growth may change when stomatoregulators are applied to leaf surfaces. If the stomatopores close completely, this will stop photosynthesis. However, if stomato openings are only reduced in size, transpiration will be reduced, but some photosynthesis will continue. Line 25. The third type of antitransparents are reflective compounds. These work by adding a white coating to leaves. This changes the amount of light that is reflected by the leaf surface. The coating is only applied to the upper surface of the leaves, so does not block the stomatopores. Coating a leaf with a reflective chemical can reduce leaf temperature, transpiration rate, and also increase the efficiency of water use. Experiments show that transpiration rate can be reduced by up to 30% and leaf temperature reduced from 40 to 30 degrees Celsius after the coating of reflective compound is applied to the leaf surfaces. Crops depend on photosynthesis for growth and final yield. Therefore, it is unlikely that currently available antitransparents would increase the yield of a crop unless the crop suffers from a low water supply or very high rates of evaporation. Spraying film-forming or stomatoregulating antitransparents on a field-grown cereal under drought conditions produced a grain yield increase of 17%. Experiments also show that any effect that antitransparents have on the mineral supply to the leaves have little effect on plant growth. Explain why plants that have been dug up and transported are at most risk of drying out. This is because the root hair cells that are supposed to absorb water could be broken or damaged during the process of digging them up, and these are going to be exposed. So, the plant cannot absorb any more water, yet water loss still occurs by transpiration. So that is why they are at a risk of drying up. Down here they say, the photograph shows some stomata on a leaf surface. If we look clearly, there is 1, 2, and 3 only. They say use the photograph to determine the number of stomata per millimeter squared on this leaf surface. So here we see 3 stomata in that specific area. The area is going to be 100 times 100, which is 10,000 micrometers squared. But they want us to find the answer in millimeters squared, so I'm going to convert that area to millimeters squared. So 10,000 micrometers squared is going to be 10,000 times 10 power negative 6 millimeters squared, using this conversion here. So this is going to equal to 10 power negative 2 millimeters squared, which is the same as 0.01 millimeters squared. If this area contains only 3 stomata, it means 1 millimeter squared is going to contain 3 divided by 0.01, which is 300 stomata. Therefore, there is going to be 300 stomata per millimeter squared, and that is going to be the answer. Moving on. In part C, explain why stomatoregulators reduce photosynthesis. Remember they talked about the idea that these reduce the stomato size. So if the stomata size is reduced, this will lead to less CO2 being absorbed, so there will be less photosynthesis occurring. And in part D, explain why reflective compound should only be applied to the upper surface of the leaf. The upper surface of the leaf is the one more exposed to heat. The lower surface of the leaf is the one with more stomata. Since this reflective compound adds a white coating, it's not a good idea to apply it to the lower surface of the leaf because that will block the stomata. So I said stomata are found only on the lower leaf surface. Most light absorption occurs on the upper leaf surface. So adding a reflective compound to the top leads to less light absorption, but CO2 absorption is not affected. So this means the plant is still going to be photosynthesizing because it can still take up CO2 at a lower temperature so that it doesn't dry out. Next they say, explain why reducing the leaf temperature reduces transpiration rate. This is about kinetic energy. The lower the temperature, the lower the kinetic energy. The slower the water molecules, the lower the transpiration rate. So I said, decreasing the leaf temperature decreases kinetic energy, so water molecules move slower and their evaporation from the leaf surface is going to be decreased. In part A, describe the role of one named mineral ion in plant growth. Common mineral ions are going to be nitrate, magnesium, phosphate, and so on. But I chose a nitrate. So nitrate because it is used in amino acid synthesis, and this can be used in making proteins. Some of these proteins are going to be for growth, others are going to be like enzymes, others are going to be growth regulators, and others are going to be structure compounds. If you chose magnesium, talk about chlorophyll formation, light absorption, and then the rate of photosynthesis being increased. In part F, describe how the water is transported from the soil to the leaves in plants. The root hair cells have a higher ion concentration, so they will absorb water by osmosis. Water is going to go through the whole plant by osmosis, moving from a region of higher water potential to a region of low water potential. So I said root hair cells contain a lower water potential than the soil, so water moves out of the soil into the root hair cells by osmosis. The water then moves into the xylem tissues and it moves towards the leaves in the transpiration pool due to the evaporation of the water from the leaves. So this brings us to the end of question 1. Let's continue to question 2. Question 2, the diagram shows a section through a human kidney with some structures labeled. So they say which structure is labeled P in the diagram. Structure labeled P is going to be the nephron. So here the answer is a B. And then they say which fluid is carried in tube S. Tube S carries blood. And the next they say which tube is labeled Q in the diagram. Tube Q is the ureter. It takes urine from the kidney into the bladder. So the answer here should be a C. In part B, analysis of the composition of urine can be used in the diagnosis of problems with kidney function. Urine samples are taken from 3 patients and the results are shown in the table. So we have these 3 patients. So the problem with patient W is the basement membrane where ultrafiltration takes place. And the problem with patient X is the proximal convoluted tubule because glucose was not reabsorbed 100%. Discuss which processes in the kidneys may be affected to produce the results shown. Include in your answer where in the nephron the processes occur. So for patient W, ultrafiltration is affected. This is because they have proteins. Proteins are larger molecules and they shouldn't go through. Filtration from the glomerulus into the Bowman's capsule allows proteins to pass through the basement membrane. So for patient X, since urine contains glucose, selective reabsorption does not occur properly. The proximal convoluted tubule does not reabsorb 100% glucose into the blood. And for patient Y, glucose is absent, but they have very high amounts of water. There could be a problem with their collecting ducts. So I said for patient Y, reabsorption of water by the collecting ducts is affected. The membrane is less permeable to water. Moving on. Here they say, describe how to test for glucose in a sample of urine. This is by using Benedict solution, and then you heat. If a color formed is red, orange, yellow, or green, then this confirms presence of glucose. So this brings us to the end of question 2. Let's continue to question 3. Question 3. Scientists study the population of plants in 2 fields. There is field X and Y in southern England. Investigated the numbers of 3 different wildflower species, A, B, and C, so that they can compare the biodiversity in each field. So this is A, that is B, and that is C. They say the table shows the numbers of plants of each species per meter squared counted in each field. So we can see field X, field Y, and the different species that are present. We can see in field X, species B is the lowest abundant one, and you can see there is not much evenness. There is more evenness here as well as here than that, because some have 3 and some have 13. The distribution of species B in field X is not even. Then when we go to field Y, we can see species A is completely absent. This is the most abundant and it looks evenly distributed. In comparison, we can say field X has a higher biodiversity than field Y, based on the information given to us because there is no species A here. So here they say calculate the mean number of plants of species B in field Y. So we have to add that, and then divide by 3. I get 23 per meter squared, and this is my answer here. Moving on. Here they say, describe how the scientist could determine the number of each plant species per meter squared in each field. So just use a quadrat. You have to throw randomly at random coordinates generated using a random number generator, and then count the number of each species in the quadrat, then divide the number obtained by the area of the quadrat. Because remember, per meter squared meaning per area, so just get the number per area of quadrat, you'll be able to get the number of organisms per meter squared. And in part B, discuss what the results show about the distribution of species in each field and the difference in biodiversity between the two fields. This is similar to what I explained when I was going through the table. So we can see field X has 3 species, A, B, and C. Species A is the most abundant in field X, and field X has less variation in the numbers, so there is even distribution of species A and B. Also, field X has greater biodiversity than field Y. The reason for this is in field Y, species A was missing. However, only 3 counts per species per field were made, so the results are limited to be conclusive. The last point is about the idea that they just made 3 counts per field. The number of counts are not many for us to make a conclusive judgment on the abundance and distribution of organisms within this specific area. So, moving on. In part C, name an abiotic factor that could affect the distribution of the plant species. This is quite easy. Temperature is an abiotic factor, sunlight is an abiotic factor, water, mineral ions, and so on. So any of these would award you 1 mark. So this brings us to the end of question 3. Let's continue to question 4. Question 4, the diagram shows part of a molecule of DNA, which is that. They say what is meant by the term genome. The genome means all the genes present in an organism. The next they say describe the differences between the structure of DNA and the structure of RNA. DNA is double-stranded while RNA is single-stranded. DNA contains the sugar deoxyribose while RNA contains the sugar ribose. Then DNA contains base thymine while RNA has no thymine. Also RNA contains base uracil and DNA does not contain that. Moving on. In part C, describe the roles of RNA in protein synthesis. In protein synthesis, we talk about messenger RNA. Messenger RNA is going to be produced through transcription, and the transcribed messenger RNA is going to contain a copy of the genetic information from the nucleus. It carries it to the cytoplasm. In there, it binds to the ribosome for translation to begin. Transfer RNA, containing an anticodon that is complementary to the codon on messenger RNA, brings amino acids for the polypeptide to be formed during translation. If you want, you can talk about the idea that this is a condensation reaction when the polypeptide is being formed. So this brings us to the end of question 4. Let's continue to question 5. Question 5, the diagram shows part of the female reproductive system with some structures labeled. So we have structure A, which is the oviduct, we have the ovary, we have the cervix, and then we have the uterus. So they say name the structures labeled A, B, C. We have the oviduct, we have the ovary, and then the cervix. Then in part B they say, describe the role of structure D in reproduction. D is the uterus. The uterus allows for the embryo to implant, and it also allows for the formation of the placenta so that fetus can be supplied with oxygen and nutrients, and then so that waste can be removed. So I said D is where implantation of the embryo occurs, and in D, the uterus is where the placenta grows to provide oxygen and nutrients as well as removal of waste. Moving on. In part C, the graph shows the changes in the level of hormones released from structure B during one menstrual cycle. On the vertical axis, we have hormonal levels, and on the horizontal axis, we have the day. There is hormone M that increases before day 14, and there is another hormone N that increases after day 14. Day 14 is when ovulation occurs, so this hormone here is going to be progesterone, and its purpose is to maintain the walls of the uterus so that they do not shed away in preparation for pregnancy. The hormone M is going to be estrogen. Its purpose is to cause the growth of the uterine wall. So here they say explain the importance of the changes in hormone M and hormone N during the menstrual cycle. I said hormone M is estrogen. The levels of this hormone increase and peak before ovulation. This causes the uterine walls to grow, meaning be repaired, and it also stimulates the release of LH hormone while inhibiting the release of FSH. FSH is follicle-stimulating hormone, and this is luteinizing hormone. Also, hormone N is progesterone. It increases and peaks after ovulation. This hormone maintains the uterine lining. When progesterone decreases, the endometrium will break down and menstruation will occur. Moving on. Here they say other hormones are involved in the menstrual cycle. Describe the role of one other named hormone in the menstrual cycle. Of course, there is luteinizing hormone and follicle-stimulating hormone. The role of follicle-stimulating hormone is to cause the egg to mature inside the follicle, and the role of luteinizing hormone is to cause egg release during ovulation. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Measles is a disease caused by a virus. The graph shows the number of cases of measles each year and the percentage of 5-year-olds vaccinated between 1950 and 2016 in the UK, percentage of 5-year-olds vaccinated, and then the number of cases of measles. On the vertical axis, we have the number of cases of measles, and on the horizontal axis, of course, we have the time in years. The other side, we have the percentage of 5-year-olds vaccinated. We can see the measles vaccine was introduced about here. After introduction of the vaccine, the cases go down as the percentage of 5-year-olds that are vaccinated increases. So down here they say, determine the difference in the percentage change in the number of cases of measles between 1950 and 1965, and the percentage change in the number of cases of measles between 1968 and 1983. I began with 1950 to 1965. In 1950, we see the cases are 420, and in 1965, we can see the cases are going to be 520. So 520 minus 420 divided by the original, which is 420 times 100, giving me 23.8%. This is an increase. And then 1968 to 1983, we're going to come to 68, which is that, and we have 490, and to 83, which is that, giving us 120. So 120 minus 490 divided by 490 times 100 gives us negative 75.5%. There is a decrease here. So the difference between these two is going to be 99.3. Moving on. Here they say, explain the relationship between the number of cases of measles and the percentage of 5-year-olds vaccinated. They want you to use the information from the graph in your answer. If we go back to the graph, we can see after introduction of the vaccine, the numbers of 5-year-olds being vaccinated increased, and as it increased, the cases of measles began to decrease. And about 1998, we can see the cases are almost zero when the percentage of vaccination is higher than 90%. So I said, before the vaccine was introduced, the cases were high and more variable than after the vaccine introduction. This is because there was no immunity or antibodies among children. But after introduction of the vaccine, more children became vaccinated, so more children had immunity or they produced memory cells, and the reproduction rate of the virus decreased. Also, there were no cases in 1998 when the percentage of vaccinated 5-year-olds was over 90%. And down here they say, some children are receiving treatments that reduce the activity of their immune system. Explain why vaccinations are not used on those children. Because the activity of their immune system is reduced, it means even when vaccination occurs, the immune system is not going to be active enough to produce antibodies as well as memory cells, so they will not have any protection. So I said, since their immune activity is reduced, they can produce sufficient antibodies. Also, no memory cells are made, so secondary immune response can be achieved. So this brings us to the end of question 6. Let's continue to question 7. Question 7. The following passage contains information about using micropropagation to produce plants. They want you to complete the passage by writing a suitable word in each blank space. This is for micropropagation, where explants are used. Micropropagation uses small fragments of plants which are regrown into whole plants. These fragments of plants are known as they are explants. The surface of each fragment is sterilized to prevent growth of course, microorganisms. The fragments are placed in growth medium. The growth medium contains agar and a source of energy such as glucose. This method is able to produce large quantities of genetically identical plants called clones. One advantage is that micropropagation is quicker than sexual reproduction, which involves flower and seed production. Another advantage of micropropagation is that plants can be produced throughout the year. So this brings us to the end of question 7 as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.